Yeah, today this conference is going to be about fibrillarin and phosphoinosite. Fibrillarin is a methyltransferase that is located into the nucleus, cell nucleus, in most of the organisms from eubacteria all the way to humans. Phosphoinosites are some type of lipids, particular lipids. They are mostly studied as second messengers, but today I will be telling you a little bit more of a different story as to what we have been working on the last few years. But to understand fibrillarin, we have to understand the nucleolus, where is the principal area where this protein locates. The nucleolus in animal cells and in plant cells, they differ quite significantly in shape and in components. The plant nucleolus has a center, a vacuolar center, while the animal resides these particular areas that are dark, which are the DFC, FC, and GC. GC is where the ribosomes are assembled. FC and DFC is where the ribosomal transcription is taking place. And let's not forget that for all the transcription that is carried out in the cell, the ribosomes are required for this transcript to be useful in any form. So the regulation of the translation machinery is essential for all the transcription later on to be able to take place for the later proteins. Now, the compartments, the dense fibrillar compartment and the fibrillar center, these are the main parts where transcription is taking place in the cell. And this ribosomal transcription is regulated differently than POL2 transcription. POL1 transcription is regulated by the number of sites that are always being shot at maximum speed. So POL1 transcription is always being fired at the maximum speed in every part of the promoter. However, unlike POL2 transcription, POL1 transcription has several hundreds of them. So you can regulate the level of transcription in POL1 transcription by reducing or increasing the number of sites that you have to fire. And so it's a very different mechanism of regulation. But what about fibrillarin? Well, fibrillarin, as I mentioned, is a methyltransferase. This is the enzyme that is in charge, or is well known to be in charge of the methylation of the RNA, the ribosomal RNA, in more than 100 positions to be able to form the ribosomes. Okay, so the proteins that conform the ribosomes are attached with this RNA, and this RNA is the main enzymatic activity. The enzymatic activity from the ribosomes is not carried out by the proteins, but by the RNA, and this RNA has to be properly methylated. When you have alterations in the methylation, then you get alteration into the translation, which leads to some types of cancer, and it can be having other type of translation effects that may have other various sicknesses. Organisms, like in plants have from 1 to 3 different genes that code for fibrillarin. In Arabidopsis, it has 3 genes. One of them is inactive, which is fibrillarin 3. The other 2, fibrillarin 1 and fibrillarin 2, they express at a high abundance in different tissues. And I will show you a little bit as to some of the differences biochemically between both of them. In humans, the fibrillarin mostly is being studied one of the genes. However, if you go into the gene banks, you find that there's a second gene that is rarely ever published about, and it does have some effects and some transcription, especially in the brain. The whole structure for fibrillarin mostly it has been studied in a complex. This complex for the methylation of ribosomal RNA, it requires a few other proteins like NOP5, NOP7, 15.5, and this is the main way as to how the fibrillarin is known to act. However, there are other possible scenarios in which fibrillarin is used. As an epigenetic, fibrillarin is not only able to methylate RNA, but it can also methylate histone H2A. We and other groups have published this part, and this works together with a FAC complex to be able to regulate the positioning and transcription of POL1. So this is completely different from what is typically known of fibrillarin as a simple methylation for ribosomal RNA. It is also interacting with some of the POL1 machinery. RPA49 is one of the components of POL1, and fibrillarin interacts with this protein. It also interacts with the cellulation machinery of GAR1, and I will explain much later as to how do I think this is happening. But between fibrillarin and GAR1, they are the ones that are in charge of methylation and cellulation of the ribosomal RNA. Later, it is also involved into the processing. These RNAs, the ribosomal RNAs, have to be cut into different pieces and has different sections and positions where this RNA has to be broken. And this is one of the main parts of this study that I'm going to be talking about. And finally, the translation. It is unknown until today as to what is fibrillarin doing interacting with some of the machinery of the ribosomes for the translation process. So one of the things that we found early on was that fibrillarin can also interact with a family of lipids, these phosphoinosites, PIP3, PIP4, PIP5, phosphoinosite at the position. The numbers indicate the position in which is phosphorylated. And this whole family of phospholipids are involved, are very well known as second messengers, in which the particularly PIP2 or PIP4.5, it's broken down and it creates a whole cascade of events from the cytoplasm all the way to the nucleus. And we found that the fibrillarin in plants, the fibrillarin 1 and fibrillarin 2 have a very different pattern in which they actually interact with these phosphoinosites. Also, the human fibrillarin can interact particularly with PIP45 and PIP34, as well as PIP5. So these were unknown questions as to what are these phospholipids doing and how could they actually be inside the nucleus. So we set up to make some of the tools to be able to see the lipids themselves, as we can see them here in red in immunolocalization. We developed this tool that is able to, a protein domain that interacts with the phospholipids, particular phospholipids, and in this case is PIP2. And in green, we can see the fibrillarin. This is the whole cell nucleus. If you wash it with Triton, Triton removes all the unbound lipids. So you no longer see the membrane lipids, but you only see whatever is stuck and is interacting with proteins. So these are lipids that are actually bound to proteins. And what we found is within the nucleus themselves, we can see this colocalization with fibrillarin, as well as with UBF. UBF is one of the main transcription factors for POL1 transcription. And we carry out some of the path plots with the recombinant proteins to see that it actually interacts, as fibrillarin and UBF, both of them interact. OSH1, which is a PIP4 binding, it interacts with PIP4 and not with PIP2. And non-specific protein IMP5, we could see that it doesn't interact with any. And we did also pull downs to make sure that the lipids binding to these proteins were real. Then we go to a little bit of the next story before I can actually join both of them. And that was while we were producing the fibrillarin for these studies, the recombinant fibrillarin, we found that it was having some strange effect. The first studies were done to see whether if the fibrillarin interacting with the RNA, as I mentioned, it has to interact with a particular RNA before it can interact with the ribosomal RNA. So this non-coding RNA, usually U3, but it can interact with about 100 different type of small non-coding RNAs. We were producing some of them, and along with that, they were producing other RNAs that we were extracting. And when we tried to add the fibrillarin, what we saw it was degradation. And at first we thought it was probably contamination of some of the RNases. And it took a long time to try to purify as much as we could the fibrillarin, remove all the possibilities, and until we actually made some discovery that it was actually the fibrillarin itself that was able to cut the RNA. We tried different type of experiments of the time with different components, salts, and we also tried within the cells using as a positive control RNase A. If we see the DAPI and pyrimidine gamma, we can see the RNA. So we can see how the cells are surrounded by RNA, especially in the cytoplasm. Upon treatment with RNase A, it removes all of the RNA visible. Additional fibrillarin does pretty much the same. It leads a little bit more within the cell nucleus, the nucleoli. And we also tried the GAR domain, which I will explain a little bit more. So one of the first things was what would be the lipids in action with this activity. And to surprise, some of the lipids have some effect into changing some of the pattern of the digestion, and some of the lipids actually inhibit this activity for the fibrillarin. And we decided to try different amounts to figure out what would be the conditions, and particular amounts, the small amounts of the lipids were able to activate the fibrillarin to be able to cut specifically. Large amounts of the lipids basically result on the opposite, inhibits the activity. And this we'll find out a little bit more to understand this later on. But we also looked as to what was the part of the fibrillarin itself that was responsible for this enzymatic activity. So the typical experiment is to chop the protein into the different domains that it has. We found that the GAR domain is the one that is responsible for the activity, unlike all the others. So this GAR domain, we could actually use to be able to cut RNA within the cells, or in gel experiments. We tried this to be able to prove that it was the fibrillarin itself, in which the gel itself is loaded with a lot of RNA. We run the protein, and you can actually see where the band is digesting. So we figured out that it was the GAR domain, and we decided to also try within the complex itself. One thing is the protein, but another thing is the whole complex of NOV58, NOV56, and protein 15.5 to see whether if that have an effect on the digestion. The whole complex, the native complex, has almost no activity, but has no activity if you remove the GAR domain. So if to the fibrillarin you remove this GAR domain, then it doesn't have any effect. But if you leave the GAR domain, it can actually cut the RNA. Another, besides PIP5, we tried PIP2, and PIP2 seems to do the opposite, basically inhibit the digestion activity of the fibrillarin complex. So we decided to look a bit close, because before we only did confocal microscopy, and in confocal microscopy, it looked like PIP2 and fibrillarin were actually colocalizing. However, upon doing scene imaging super resolution, we found that actually fibrillarin goes around and forms a special capsule around PIP2, where the red dots are PIP2. The nucleolus is the void of this phosphoinosite, and the green parts are the fibrillarin, and it basically just surrounds the PIP2. So the activity itself has to be modulated in some way by the phosphoinosite, but it doesn't really have to be the concentrations are not so simple to measure because the lipids themselves form these particular structures. We tried, therefore, to see whether we could actually alter these structures by mutating this GAR domain in particular positions. So we affected this GAR domain and we changed the dynamics in which these the shape of these clusters were done within the nucleoli, as well as the dynamics in which they actually move. Also, they have different effect on the digestion on the RNA. The single mutations, one of them increases the speed in which the RNA is degraded. The other one actually prevents the RNA from degradation. So that was another hint that the story of the protein being able to digest RNA was actually correct. We therefore set up to try to investigate more about this GAR domain. This GAR domain is actually found in only a handful of proteins, with all of them that interact with RNA and are found within the nucleus. Nucleolin, one of the most prominent, but a few other ones like GAR1, which actually interacts in a similar process as fibrillarin. All of them have this GAR domain, and this GAR domain, when we looked upon that, we also expressed from different proteins, and we found some of them, as well as from, unlike the one from TGB, which doesn't have any activity. But when we began to think as to the localization on these lipids within the nucleus, we found all these different particular position metabolism within these lipids that changes from one form to the next, and they seem to have the capabilities of aggregation. So this lets you to the phase separation, and this phase separation is an interesting process in which we could create structures without the use of much of the energy. Let us remember within in the cell nucleus, we don't have, inside the cell nucleus, we don't have any membranes that will be able to encapsulate particular processes. So we need somehow to be able to aggregate or concentrate particular proteins or particular components to be able to have a particular function. And as we have published before, PIP2 is also involved into POL2 transcription and some of the lipid islands that are formed within this cell nucleus. But looking at these structures, we were set up to find out as to whether fibrillarin also was part of this, and the GAR domain was involved, as well as other proteins that have similar motifs. And this lets you as to how you can actually create aggregates, in this case like the nucleoli, in which you form a large number of compounds together. And then if you change the metabolism of the lipids, you can dissociate these aggregates and to be able to reduce them. So it also lets you this trafficking of molecules to be able to move from one place to another with a low energy requirement. One of the characteristics of this GAR domain, as well as some of the proteins that we were looking, is they have a protein disorder. And we found that a lot of the proteins that have that are disorganized, that do not follow any of the alpha or beta shield conformations, they tend to interact with some of these lipids. So this disorder is actually quite interesting in terms of some of this assembly of complex structures that can be found within the cell nucleus. And we looked at a few other molecules and figured out more or less as the model as to how this protein is working. So what we're actually looking more is as to how these structures or structures within the cell nucleus are maintained within these lipids with fibrillarin and actually nowadays also RNA. one of the particular reasons why I came into this project, I was not so much interested into methylation of ribosomal, but I was interested by the fact that when you mutate or remove fibrillarin from the cell nucleus, the structure of the nucleus is altered. So why would a methylase alter the whole structure of the nucleus? And one thing that we began to see is that it could probably be involved into the processes like this, in which it requires for some of the processing of the RNA. When it interacts with this U3 RNA, it doesn't digest U3 or small nuclear RNAs, but it digests other parts of RNA. So it might be a way to target a specific RNAs to cut at specific locations. And this might be an interesting process, especially during the mitosis mechanism, in which all these structures have to be disassembled to form, to be able to allow for the cell to divide. So that was part of the talk that I wanted to have for today. And I want to acknowledge some of the people from my lab and some of the people that I collaborate, especially with Pavel Hosak, Milada, and Nicolas Vitale from Czech Republic. Thank you. I don't know if you guys have any questions, and I'm free to answer any questions. You can just send me an email. I'm sure it will appear within this brochure. So thank you again.